Thank you very much, Rodrigo, for the introduction. This is Montevideo. This is one of the most beautiful places of our city at the Rio de la Plata, and that's why I select this picture to show you something from our country. First of all, something that Rodrigo said, I would like to thank the Programa Fondo de Atracción de Académicos Internacionales, which was supposed to be started in 2019, but at the beginning of 2020, but the pandemic delayed the start of the program, so it's a pleasure to be part of this program and aid at the program of postgraduate at the University of Talca, the Doctorado in Ciencias Biomédicas, and again, and also would like to thank my colleagues here from the Centro de Investigación en Trombosis, and Eduardo Fuentes, Iván Palomo, and the group, where we are being collaborated for such a long time. This is, I think, my 5th time here in Talca, but I received many students in my lab, so it's a great collaboration, and I hope this stay this month could enhance the collaboration between our lab and the University of Talca or other labs in Chile. For my talk, one of the things that I want to mention, to say is, during the inflammatory process, we have signaling actions and injury, and depending on which side this part we are, we can have the activation of signaling pathway by some compounds, in our case, the acid, and we'll explain later what they are. But when the oxidative stress is really high, we obtain an injury in the cells, for macrophages, that lead to damage, mitochondrial dysfunction, apoptosis, and diverse inflammatory processes. So we started 10-15 years ago in our lab to study, apart from lipid oxidation, what we call lipid nitration. Lipid nitration is also oxidation, but it's different than lipid oxidation on the products that we form. If we talk about only lipid oxidation, the products will be lipid hydroperoxides or isomerized fatty acids, but depending on the oxygen levels, this unsaturated fatty acid, which has several double bonds, can suffer the formation of these compounds, nitro-fatty acids, where nitrogen oxide, which is a free radical that can be formed, for example, from peroxynitrite, or in the stomach at acidic conditions in the presence of nitrate, we can form this compound that lead by several reactions to the formation of this nitro-fatty acid. And I put this square in the nitroalkenes because these compounds, where the nitro group is bonded to the double bond, give to this compound several important anti-inflammatory and protective actions, and particularly because when the nitro group is bonded to the double bond, makes the compound electrophilic, and the capacity of being electrophilic forms this product for being signaling compounds. That is, when you need these compounds to activate some pathways, it can activate, and we will see that it can be reversible, and when we don't need more, that the activation of that pathway will reverse the action. And that is why we, in the rest of the talk, when I talk about nitro-fatty acid, always will be nitroalkene and not these products, which are not electrophilic because the nitro group is bonded to single bonds. Okay, which are the nitrating species involved in the formation of these products during inflammatory processes? As I told you, from peroxynitrite, it can be formed nitrogen dioxide, from the monoperoxycarbonate also say, different enzymes like myeloperoxidase can form in the presence of hydrogen peroxide and nitrite, nitrogen dioxide, and this is what happens in the stomach. And all of these sources of nitrogen dioxide can be examples of the formation of these radicals that can lead to the formation of nitro-fatty acids. Which are the reported actions of these compounds, both in vitro and in vivo? We have activation pathways and inhibition pathways. For example, I showed today about the production of superoxide from pH oxidase, but also it can reduce vasodilation dependent on epoxy hydroxylate. But for inflammatory processes to induce the formation of antioxidant enzymes like hemoxygenase or some enzymes necessary for the synthesis of glutathione and increasing the reaction potential of the cells, nitro-fatty acid activates the NRF2 KEAP1 pathway, releasing NRF2, leading that this nucleus these antioxidant enzymes. And also, in our lab, we demonstrate that the nitro-fatty acids are able to inhibit the activity of cyclooxygenase and the formation of some prostaglandins that could exert the levels of nitric oxide that would be a relevant player in some inflammatory processes, in particular when, for example, an infection occurs. In this presentation, I will focus on nitration of arachionic acid. Okay, this is, as you can see, there are several different nitric acid, but in my lab, in my group, we focus on nitration of arachionic acid, and we obtain, as marked in red, 4 different isomers. That means that the nitro group is bonded at different carbons at the double bonds. And why we decided to study arachionic acid? Because the arachionic acid pathway is relevant for inflammation. Why? Because arachionic acid, when released from the phospholipid at the membrane, can form prostaglandins, can form the substrate for the different prostaglandins. For example, thromboxane A2, which is a pro-aggregant compound in platelets, or even by lipoxygenase, the formation of some compounds relevant for the physiology of platelets and leukocytes. And again, as explained today by Professor West, in macrophages or in other cell types, is the precursor of prostaglandin E2. So, what we suggest that the formation of nitroarachionic acid can divert this product, this substrate, from these normal pathways to form this compound that has more activities, new activities and protective activities compared to its precursor, non-nitrate. Some previous work from our lab, and this is a slide that shows part of the work that we have done in the last year in the lab. We observed that in platelets, nitroarachionic acid can affect the activation of PKC, reducing platelet activation by this mechanism, can reduce the formation of thromboxane A2 by inhibiting the activity of COX-1, but also in endothelium, it is able to modify the capacity of activation of 1-elate cyclase and the capacity of exert smooth muscle cell relaxation. So it can be a protective molecule, but in this case, I am showing just in these 2 times. But as said previously by my colleague, we are in a summer school of inflammation. So why we started to study the effect of nitroarachidonic acid during inflammatory processes, and in this case particularly in activated macrophages, because activated macrophages express COX-2, the inducible form of cyclooxygenase that is increased during inflammatory processes. Also because they have lipoxygenase that can oxidize arachidonic acid, and also because during the inflammatory processes, superoxide and nitric oxide are formed and are necessary for the activated macrophages to exert the activity during inflammation. This is what I told you before, nitroarachidonic acid, by its capacity to form covalent bonds due to its electrophilic capacity with KIP1, is able to release NRF2. NRF2 migrate to the nucleus, induce the expression of antioxidant enzymes. We show that the nitroarachidonic acid inhibited the here it says prostaglandin, the cyclooxygenase isoform 2, but it's 1 and 2, and in particular, these are the reports that we published in the last year. This is the first report of a peroxidase inhibitor of the COX-2 isoform. It was a pretty good paper. Also, we are continue studying the effects on COX-2, and in this review original paper where we collaborate with Stephen Yang, we showed that using EPR we can follow the formation of different prostaglandin and the synthesis of nitroarachidonic acid due to the enzyme activity. We observed in kidney tubular cells activated by angiotensin II that the compound prevents mitochondrial dysfunction and also induces the expression of these antioxidant enzymes. And finally, in ALS, amyotrophic lateral sclerosis, which is a neurodegenerative disease, our compound was able to activate NRF2, and that protects the cells from being from the dye exerted due to the disease. Okay, but go now to what happened with the production of superoxide. Superoxide formed by NADPH oxidase, which is necessary in activated macrophages to exert activity when bacteria infect the cell or when the cells are activated during an inflammatory process. And this was evaluated by different methods in our lab. This is the mechanism of activation of the NOX2. NOX2 is a complex of different subunits that need to be assembled correctly at the membrane to produce superoxide from oxygen using NADPH subunits. And these cytosolic subunits have to migrate have to translocate to the membrane to form the active complex. And this occurs when a stimulus come to the cells, and some of these subunits have to be phosphorylated to perform the correct assembly of the enzyme. So, to analyze if our compound was able to affect the production of superoxide by NADPH oxidase, we evaluate the production of superoxide, the final product, by different techniques like cytochrome C reduction, complex red fluorescence, chemiluminescence, and by flow cytometry using this compound FC oxyburst, but also by Western blot by looking at trying to identify the mechanism of action of this compound. This is cytochrome C reduction studies. As you can see here, we activated the macrophage with PMA. The non-nitrated arachidonic acid did not exert any effect, and in a dose-dependent manner we observe a decrease in the production of superoxide, and we know that it's superoxide because when you study cytochrome C reduction, you have to perform the control with SOD. If you observe the decrease of cytochrome C reduction due to the production of superoxide in the presence of SOD, is because cytochrome C is reduced by superoxide. And the reviewer asked to show that when we have a model of knockout mice, we also analyze the formation of superoxide, and of course, if we have cells from knockout mice for NOx2, we didn't observe any changes in the cytochrome reduction. So NOx2 apparently was one of the targets of our compound, and this was confirmed by luminescence, Amper's red oxidation, and this is the flow cytometry we use in the FC oxyverse, which is an antibody bound to a BSA molecule that is phagocytized by the macrophages, and nitroarachidonic acid reduces the capacity of this macrophage to be activated, which not occur with arachidonic acid. And again, this was due to the decrease of NADPH oxidase activation, because using some inhibitors of phagosome for the phagosome formation, we obtained the same result. So it wasn't that the nitroarachidonic acid was affecting the capacity of phagocytizing this compound, but it was because it was decreasing the formation of NADPH oxidase. This is fluorescence. That's why we can measure the activation of the enzyme. And finally, we obtained this important result that the effect of nitroarachidonic acid was reversible. What it means? First of all, we don't need an activated macrophage producing superoxide for such a long time, because superoxide and reactive oxygen species can be can exert damage to the cells. So in a way, we have to increase the production of superoxide when needed, and then reverse the effect and come back to the basal level. And what we observed was that after incubating the enzyme, after activating the enzyme, and in the presence of nitroarachidonic acid, the effect was prevent the correct assembly of the enzyme. We analyzed if P47 FOX, which is one of the main cytosolic subunits that need to migrate to the membrane to form the active complex at the membrane, was migrating or not in the presence of nitroarachidonic acid. We performed Western blot experiment. The first thing was that we observed that in the membrane fraction, P47 FOX decreased in the presence of nitroarachidonic acid, but in the cytosolic fraction, it was increased. So something was preventing the migration of P47 from the cytosol to the membrane. So something was preventing the correct assembly of the enzyme. And that's why superoxide levels were lower in the presence of nitroarachidonic acid. So this was the first part of our study our mechanistic study of the inhibition of NADPH oxidase by nitroarachidonic acid, and we observed that the translocation of the subunits to the membrane to form the correct assembly of the enzyme was prevented by nitroarachidonic acid. But then we performed this microscopic analysis using a fluorescent activated macrophages, and these are the fluorescence of the compound. This is the P47, and in the presence of course, in the absence of activation. We didn't see the migration and colocalization of P47 and this compound at the membrane, but when activated in the presence of arachionic acid, we observe a colocalization at the membrane where the active complex is active, and it was different when the cells were activated with nitroarchionic acid, confirming that the migration of the cytosolic subunit to the membrane was prevented by our compound. Another player appeared in this history, and it is protein disulfide isomerase. In 2009, the group from Francisco Laurindo at the University of Sao Paulo demonstrated that PDI is a key player to regulate NADPH oxidase activity in activated macrophages. And then, 2 years later, Antonio Marcos Narváez, who was at the time a postdoc at Professor Laurindo's lab, and now he's at the University of Maranhão in the north of Brazil, and also a collaborator with the Center of Thrombosis here in Talca, showed that PDI interacts with P47 FOX, leading to a redox activation of NOX2. And with this previous data, we started to think, is PDI also a potential target for nitroarchionic acid? And the answer started to be yes, because this is PDI, which has two different active sites. And you can see here that both active sites has this diethyl motif, okay, which is necessary to participate in the redox folding of proteins through oxidation reaction and isomerization reactions. And it's important because cysteines can react with nucleophiles to be attacked by the nucleophilic part of the nitroarchionic acid. And the nitroarchionic acid can modify the cysteine, it can inhibit the PDI, and then prevent P47 migrating the membrane and activating NADPH oxidase. This is the studies performed from one of my PhD students, Lucía González, in Uruguay. She started to evaluate the capacity of nitroarchionic acid to inhibit the enzyme PDI, and we observed here that in the presence of nitroarchionic acid, there was a decrease in the activity of the reductase enzyme, which was dose dependent, but arachionic acid alone and in the presence of the nitroarchionic acid, but without enzyme, the technique, which is the precipitation of insulin, was not affected. So this decrease in the activity was due to the presence of nitroarchionic acid. Also, the chaperone activity of the enzyme, which is necessary when proteins are being folded for the correct formation of proteins in the cells, was also inhibited, but at this time, 10 micromolar reduced almost 30% of the activity, but it was really nice to see that there was a decrease in this activity, because the chaperone activity is in a different part of the enzyme. The chaperone activity is here, not in this active site. So something was also occurring at the folding structure of the protein, and I will show you later what was going on. And the final study to decide that our result was due to the covalent modification of suggesting it was a covalent modification of the protein, was this study using nitric oxide NOx7, which did not exert any effect on the enzyme, so the cysteine were not forming nitrosocysteine, but our compound, in the presence of nitroarchionic acid, really decreased the activity of the enzyme. So at this time, we were aware that P47 was one of the targets, or can be one of the targets for the decreasing of NOx2 activation, and maybe PDI, due to the modification of the cysteine necessary for the activation of NOx2 was being modified covalently by our compound. And to evaluate this, we performed this. This was really a nice experiment. We performed this Western blot where we using 5,000 to modify the protein. We obtained that in the presence of nitroarachidonic acid, there were an estimative of 2 to 3 addition of pegylated nitroarachidonic acid. Of course, mass spectrometry is necessary to confirm this data, and we observe by spectrometry that this is the enzyme, and this is the enzyme incubated with nitroarachidonic acid, and we obtain, after incubation, the presence of 3 peaks, each of them with a shift in 348 Daltons, which is the mass of the nitroarachidonic acid. So our compound was really covalently modifying the enzyme. Okay. This is to show that each cysteine can be modified. So our compound was really modifying the enzyme, so it was a target. But what happened when we use activated macrophages? Was PDI being affected or something, or it was because it was being inhibited cells because the former experiment were with the activated enzyme? And we obtained these pictures that were showing PDI inactivated cells. PDI goes to the membrane, and maybe because it's interacting P47. But when we activate the macrophages in the presence of nitroarachidonic acid, we started to see these bubbles, which depended on the time, full of PDI. But those bubbles remain on the macrophage. So a change in the morphology of the cell, but those bubbles were full of PDI, but were not secreted. These bubbles, like the protosomes, because of the presence of actin, that showing that these bubbles were protosomes. And again, apart from remodulating the structure of the cells that you can see here, this is the activated cells, and in the presence of nitroarachidonic acid, actin is being modified. We didn't observe a colocalization of PDI with those actin, saying that or suggesting that these bubbles were not protosomes. And you can see here in a big picture that PDI is in these bubbles that not merge with actin. So we don't know if those vesicles or those bubbles are being released later, like in exosomes or other type of vesicles. We are studying what is going on there, because as here we know that PDI inactivated macrophages co-localize with P47, but not in the case of the macrophages activated with nitroarachidonic acid. Finally, to see what was going on with the NOx production, when we activate macrophages with LPS interferon gamma, we observe an increase on the inducible form of NO that was the same in the presence of arachidonic acid, and was really decreased in the presence of nitroarachidonic acid. From previous work, we know that this is not at the transcriptional pathway, so we don't know exactly which is the mechanism yet, but we are studying that. But the important thing was that COX-2, the induction of COX-2 remain the same in the absence or presence of nitroaconic acid when activated, when we activated the macrophages. So we know that the enzyme is can be inhibited by the compound, but its transcription is not affected. And the current work is studying the mechanism of inhibition of COX-2 by nitroaconic acid. This is our previous work in COX-1, when our proposal is that the inhibition of the enzyme, which is a heme protein, is the release of heme producing an inhibited enzyme. So we obtained, in a collaboration with Professor Marnet in Vanderbilt University, mutants for COX-2, mutants for some residues at the COX active site and at the POX active site, and we observe differences in those mutants. I'm not going to be deep because of the time in this, but the thing is that the way that the product enters the active induce a change in the conformation of the protein, inducing the releasing. If this enzyme doesn't have heme, it's inactive. So something is going on that it changed the folding of the protein, inhibiting the enzyme. So to finish, the message is to take home. Nitroaconic acid modulates inflammatory processes by regulating the activity of key enzymes, NOX2, PDI, and cyclooxygenase. The nitroalkene affects both the reductase and chaperone activity PDI, in addition to modifying the PDI distribution in activated macrophages. Nitroaconic acid decreases the glycosylation use of mucimol, which is an inhibitor of PDI, and the inhibition by our compound nitroaconic acid was similar than the mucimol, suggesting that the effect are observed that we observed were due to the inhibition of PDI, and the mechanism of COX-2 inhibition is at the activity level and not at the expression of the enzyme under inflammatory conditions. Finally, I would like to thank my colleagues and my students in my lab at the Universidad de la República. This is a picture of our School of Medicine in Montevideo, Uruguay. Homero Rubo, Lucia González, Irene Wood, Rafael Radi. All of us are part of the Centro de Investigaciones Biomédicas. Professor Lawrence Manet from Vanderbilt University, who collaborated with us at the COX-2 experiment, and Francisco Laurindo from the University of São Paulo in Brazil, which is a long-time collaborator for PDI, and this is the foundations. Thanks. Yes. Yeah, when you the effects, the cysteines are nucleophilic. Okay, so these compounds with the nitro group bonded to the double bond are electrophilic, right? It can covalently react with this type of residues, cysteines. Okay, in the case of PDI, these cysteines are necessary for these activities. Okay, why they are reversible? Because it can change, for example, the amount of glutathione, which has a thiol that can translocate from PDI to. It's like a signaling action. It's a nitroalkylating modification which is reversible. It can be with cysteine, with histidine, It depends. The other thing is, which is important, in some type of cells, it was observed that when this nitro-fatty acid covalently bind cysteine or histidine residues, the protein migrates from the cytosol to the membrane. In this case, what I think is, if this protein is being nitroalkylated by nitroalkylic acid, it can interact with P47-FOX, and the complex cannot migrate to the membrane. That's why we observe that it's not a merge in the microscopy study.
Yes. Have you looked at the nitro-fatty acids also in the case of cancer?
Well, there are some studies from Bruce Freeman's lab in Pittsburgh. They are studying some in breast cancer cells, and they show that the other type of nitro-fatty acid can reduce the progression of the tumor in vitro models, or in some models injecting directly the compound to the tumor cells. But also we know, we and others, that these compounds are able to make the switch from M1 to M2 phenotypes of the macrophages. But in cancer cells, the only reported data is with nitro-oleic acid, which is a nitrated oleic acid, 18-1, injected directly to the tumor and reducing the progression of that. For some type of nitroalkylating, I don't remember if it is P65 from the NF-kappa-B pathway. I don't remember now, but it's something like nitroalkylating, one of the proteins that decrease the transcription factors necessary for the progression of the tumor.
The question I ask is because we have established in a immune-compromised mouse.
Mm-hmm.
There, the treatment with the nitro-fatty acids to reduce the tumor growth. But in the context of two macrophages is that you might be inducing without the immune system the tumor growth, but if you have a functioning immune system and you create two macrophages, this can be actually problematic.
Yeah.
It's like the result is fine, but easy might be not. So that's why I'm asking.
Yeah, and I think the most important thing for these compounds is that their effects are reversible, and then can act as a modulator of a pathway when it's necessary, and then reverse. And that's the main thing that's why I think the relevance of this compound. And probably in cancer cell, I'm not an expert in cancer cell, but probably what can happen is, for example, I treated platelets, for example, with the compound, and coinculate with tumor cells, and the capacity of the tumor cells to die was higher because of some signal from the platelets to the tumor cells. The target was not the tumor cell, so it was other that prevented the tumorigenicity of this one, those who has the nitro group bonded to the double bond. The nitrohydroxy derivative is the decay product of this, because when in the presence of water, for example, this is hydrolyzed, it is transformed in nitrohydroxy derivative, which it can be measured, but it's not active because the nitro group is not more electrophilic. I know I have data that denitrated compounds can be formed, but I haven't studied them deeper. Also, if the nitro group were bonded at the same double bond or not, or if it is a nitroalkene plus a nitrohydroxy. For example, this nitro group bonded here and the other one bonded here, for example. I don't know that. But probably in the case of the nitro compounds, the effect won't be the same. Perhaps because of the distribution of the electrons and the capacity of attack the cysteinols and exert it actions. But it's a very good question, and it's something that we want to explore in the future.